Okay, so we're going to start this morning with just some more practice to deal with pedigrees. It's not that bad once you figure out the few simple rules of what you should look at and how to interpret what you see. So, okay, we're going to consider 4 possible modes of inheritance. These are autosomal recessive, autosomal dominant, sex-linked recessive, and sex-linked dominant. Okay, now our approach is going to be to figure out what the answer is to eliminate the others as possibilities. So you have to kind of look at your patterns and see which ones don't work, and the one that you're left with is the right answer. Okay, so we're going to have to infer or guess by working backwards from the information that we have. Okay, so I'm going to give a few examples of how to sort of start thinking about this, and there's often more than one way to get to the answer. Okay, so here's a clicker question. Carla rules out all except autosomal dominant, sex-linked dominant, autosomal recessive, sex-linked recessive. Now you can see that Carla has the disease, and so you can say what her genotype will be in the various cases and see which one works. Okay, so for example, we consider autosomal dominant. If she's autosomal dominant, then her genotype is big A, little a. So the big A allele is the dominant one. So if that's the correct answer, she got it from one of her parents. Well, if you look at her parents, neither one has it. So it can't be autosomal dominant. She got it from somewhere, so it's not autosomal dominant. Same argument, if it's sex-linked dominant, her genotype is X big M, meaning mutant is the dominant one, X little m, the recessive one. So her genotype, she would have to get one mutant X from one of her parents. And the parent that gave her that X would show the trait. Well, neither of her parents shows the trait, so that can't work. It's not sex-linked dominant. Okay, so working to the more plausible possibilities, it could be sex-linked recessive. For her to show it, both of her X chromosomes must carry the mutation if it's recessive, so she would be X little m, X little m. She got one of her X's from her mom and one from her dad. Now, mom's not a problem because mom could be heterozygous, a carrier, but dad is a problem because dad has only one X. And if the gene is located on the X, the dad would have to show the trait since he's hemizygous. Well, dad does not show the trait, so she couldn't have gotten a mutant X from him, so that doesn't work either. So that leaves us with autosomal recessive as the only possibility. If that's true, her genotype is little a, and that means that both of her parents are heterozygous, big A, little a, because she got the mutant allele from both of them. So that works in the sense that everything else has been eliminated. So here we have the answer is C. The trait is autosomal recessive. Okay, so here's another example. I got this from our old test, the 2017 test, and it's very similar to what we looked at last time. Okay, we have to look at various possibilities and try to see which possibility works. Now, I usually like to start at the bottom for some reason. I don't know why exactly. And I usually like to start with somebody that actually inherited the trait and to try to eliminate possibilities that way and work my way back up. But that's not the only way to do it. So I would say, okay, let's start with individual 4-1, right? That's a female that shows the trait. Okay, so in order to consider sex linkage, for example, we have to say, okay, if it's sex-linked recessive, then she has 2 mutant X's, one from mom and one from dad. Well, mom is homozygous, or apparently mom is showing the trait, so she could have given a mutant X to her daughter, no problem there. But then if you look at 3-1, her father, he doesn't have a mutant X to give her. So the sex-linked recessive just doesn't work in this case. So we can eliminate that from our list of possibilities. As far as dominant is concerned, I always look for cases where people inherit it from people with non-diseased parents. So if you get somebody that has the trait and neither of their parents has the trait, well, then it can't be dominant because they got it from somewhere. So example here, and there are others as well. If we look at generation 1, individuals 3 and 4, neither one of them shows the trait, but they do have an affected son, individual 2-4. So that rules out dominance, both types of dominance really, for this particular trait. Another example is 3-2, who has the trait, and she got it from unaffected parents as well. So the trait is not dominant, and it's not sex-linked recessive. So we're kind of stuck with autosomal recessive as the actual answer here. And if we look through, we can figure out who must be carriers and who are not carriers. Anybody that has an affected parent, if it's autosomal recessive, you'd say it's little a, little a. Any affected person is little a, little a. And necessarily, they would get a little a from that affected parent because they only have little a's to give. So, for example, 2-2 is an obligate carrier because he inherited a little a from his mom, even though you can't see it. So more evidence for 2-2 being a heterozygote is that he has an affected child. So he was passed on the trait to his children. So again, you can go through the pedigree and see anybody that passed it on to their kids must be heterozygous. For example, 1-3 and 1-4 are heterozygous because they've got an affected kid, and so are 2-1 and 2-2, etc. Okay, so other affected people or unaffected people can be either big A, little a, or big A, because this is a standard heterozygous Mendelian cross, big A, little a times big A, little a. So actually 2/3 of the children will be big A, little a that are unaffected. Okay, so we've got this one down. It's going to be autosomal recessive. Okay, so the worksheet has you step through this, identifying why it can't be dominant, identify a person who rules that out. I'm sure you can do that. We know it can't be sex-linked recessive. We just did that. And then identify the people who are necessarily carriers. So I'm sure we can handle this pretty easily. Okay, so here's the one we did last time, which does also turn out to be autosomal recessive. And this is on your worksheet, and I think by now you can probably do this. I did do last time generation 4, which individual rules out both autosomal and sex-linked dominance. Well, again, anybody that gets it that has unaffected parents would rule that out. So in generation 4, you've got one affected daughter and unaffected parents. So we know that that's not dominant, either sex-linked or autosomal. Okay, part 2. I want to talk about one particular autosomal dominant trait. There's not that many of them. This is Huntington's chorea, which is a late-onset neurodegenerative disease. And it's a very unusual disease, but it's a member of a class of diseases which are called repeat expansion diseases for reasons that you'll see in a sec. Okay, so it's a progressive neurodegenerative disease, and it's kind of a sad one because there's no cure for it. And people are usually old enough that they've had kids before they know that they have it, so they can pass it on to their children without knowing about that, which is unfortunate. And death usually occurs 10 to 15 years after onset of symptoms. Okay, so initial symptoms are jerky motions, lack of control of movements, and this gets worse and worse, leading to eventually cognitive impairment and death of neurons. So this is a very sad disease. Okay, it's also kind of a weird disease from the standpoint of what causes it. Okay, it's a big gene, but the cause of it is what's called the expansion of a polyglutamine tract of the protein. So there's a stretch of the protein which has CAG, CAG, CAG in all of us, which encodes glutamine. So we all have a run of glutamines in exon, or the part of the protein encoded by exon 1 in the protein. So everybody's got a lot of glutamines in a row. This is a trinucleotide because it's CAG3. And it's an expansion disease because what happens is, or has happened at least, is that instead of having a reasonable number of CAGs, you end up with many, many more than you're supposed to have. And that leads to protein aggregation, so you're forming insoluble protein inclusions in your brain, eventually leading to neuronal death, etc. Okay, so this makes sense in terms of it being dominant, because if a protein is going to aggregate and cause inclusions, it will do that even if there's a wild type present as well. So the presence of a wild type protein can't make up for the fact that you have this aggregating thing. Now this is different from a lot of the diseases that we've seen because usually one wild type allele is enough to allow a normal function. In this case, the dominant form wins because it's going to aggregate regardless. Okay, so here's a pedigree of Huntington's being passed on. You can see males and females are equally likely to be affected. Okay, here's a diagram of the protein, actually. It says gene, but it's really a protein. Okay, so the idea is near the N-terminus of the protein, there's a CAG, N meaning some number of repeats. So a small number of repeats occurs in everybody, but some people are unfortunate in that they have a large expansion or largely larger number of CAGs, and this causes a problem with the protein aggregating. And there's some intermediate levels where these are called premutation and incomplete penetrance, and I can explain in a sec what that means. Okay, so all alleles have some number of repeats, and so all versions have CAGs there. But if you've got more than enough or too many, that leads to the aggregation, and that leads to the disease. Okay, so here's just some data, really, where people have looked at Huntington's people and examined how many CAG repeats they have on that one chromosome. Remember, it's dominant, so you only have one mutant chromosome. So everybody with Huntington's has 40 or more repeats of CAGs in that region. By comparison, people with normal Huntington have typically between 15 and 20 copies of the repeat. So a normal person has 15 or so, and you have this tremendous increase, and that's what leads to the disease. Okay, so there's a range. Normal, for some reason, is considered fewer than 28. The intermediate ones are sort of odd. The expansion idea I'll get to in a sec. But people that have 36 to 40 are kind of borderline, and so that's a later onset and less severe disease than the people with major expansions. So survival doesn't, some people will survive relatively well, whereas others don't do well at all. Okay, this anticipation phenomenon is really genuinely strange. So the idea is you have a person who's kind of borderline, maybe they've got a little bit too many repeats, but not enough to be sick, that if that allele is passed through the male germline, which is to say it's a male that has this borderline level of repeats, and then he has children, during spermatogenesis, for some reason, the number of repeats actually increases. So the child will inherit a number of repeats that's more than what the father had, and themselves will be much more affected than the father initially. So the idea is the intermediate number, which is borderline, anticipates that it's going to get worse in future generations. So this is really an unusual phenomenon. Okay, reiterating, it's inherited as an autosomal dominant because the protein is going to aggregate whether or not the normal protein is there. Okay, so we're going to move on to talk about chromosome aberrations, starting with non-disjunction today, but realize that there's many different ways in which chromosomes can be rearranged to cause problems, or sometimes not problems. Okay, so just representationally, this is looking at the level of a chromosome, not a gene. If you start off with a wild-type sequence, there's all kinds of different things that can happen to that chromosome. We've seen translocation before, where part of a chromosome gets moved to another chromosome and vice versa. You can have inversions, where a part of the chromosome basically is flipped over relative to its normal location. You can have duplication, where a part of the chromosome all of a sudden is double. You can have deletions. And for what we're going to talk about today, we can have extra chromosomes or missing chromosomes. And that, of course, leads to some serious problems. This is from an old book, but it's still an interesting idea, that if you look at fetuses that are spontaneously miscarried, many times there are chromosome abnormalities found in those fetuses, including trisomies of all forms, XOs, and other kinds of abnormalities. But even among live births, chromosome abnormalities are not in any way unusual. There are many sex chromosome abnormalities involving the X's and the Y's, lots of trisomies, and other kinds of rearrangements or changes from what would be considered normal. So these are relatively rare, but not as rare as one might think. Okay, so we're going to talk about gain or loss in the form of how you get aneuploidies out of meiosis. Okay, the important ones are going to be Turner syndrome, where it's a female with only one X chromosome, a male who's got two X's and a Y, triple X female has three X's, and of course the most famous trisomy is trisomy 21, which is responsible for Down syndrome. Other trisomies also occur, but they're much more severe and rarely compatible with life. Okay, so these arise by non-disjunction during meiosis, and that non-disjunction can occur either at meiosis 1 or meiosis 2. So what's represented here is two chromosomes that are in the form of a bivalent, although they're not shown close enough to really be a bivalent. Where what's supposed to happen, of course, at meiosis 1 is that the two homologs separate from each other, one going to one pole and the other going to the other pole. So both of the progeny cells get one of each. Sometimes that process fails. The homologs fail to separate, and both go to the same pole. And that's what's represented here. So the outcome from that is going to be, from that particular nucleus, is that the chromatids, sister chromatids, will then divide normally at meiosis 2, but that will still give you two copies of that chromosome in a gamete as opposed to the one that meiosis is trying to do. Similarly, the other gamete that didn't get any of the chromosome, they both went to the other pole, that one will be, their gametes will be absent that chromosome. So you're going to have a loss of that chromosome as a result of that aberrant event. Okay, you can also have non-disjunction at the second meiotic division, where the homologs segregated correctly at meiosis 1, but they fail to separate the sister chromatids at meiosis 2. So in that case, both sister chromatids go to the same pole in this one nucleus, and that gives you two copies of that chromosome eventually in that gamete, and the other product of that particular meiosis gives you no copy of that particular chromosome. So in this case, you're going to gain a chromosome in one gamete, and you're going to be short a chromosome in the other. Now in this case, we had normal meiosis 1, and so the other progeny will be normal. It's just this one segregating abnormally at meiosis 2. That's the problem. Okay, so here's my way of diagramming this. Looking at meiosis 1, I'm showing the paired homologs. Now what's supposed to happen, of course, is that the homologs will separate in the reductional division of meiosis, haploidizing the genome. And in that case, we then have meiosis 2, and each gamete gets one of each chromosome, which is 23. That's what's supposed to happen. If you have meiosis 1 non-disjunction, the two homologs go to the same pole, and that gives you gametes with one extra, 24 each if you're a person in that case, or 22, which is say you're missing one of whatever that chromosome is. Meiosis 2 non-disjunction is similar in that even though the homologs separated correctly at meiosis 1, the sister chromatids fail to separate at meiosis 2, leading you to have one gamete with one less chromosome and one with one more chromosome. Okay, it's important to remember that only one chromosome pair is missegregated. The others behave normally. So we're just looking at what happened to the abnormal chromosome, not the rest. But the outcome is the same. You're going to end up with either a n+1 gamete with 24 chromosomes instead of 23, or else you're going to have one short chromosome, which you're going to get 22 instead of 23. Okay, so here's a Down syndrome karyotype. You can see that the little girl has 3 copies of chromosome 21, and all the other chromosomes are normal. Okay, so trisomies, most are lethal, with very few exceptions, and monosomies in general, all are lethal except for the X chromosome situation, which is very unusual, and it has a very interesting basis for why that actually works. So you can't be short a chromosome at all, particularly, but the X chromosome and Y chromosomes have, because of our dosage compensation system, we're able to tolerate X and Y chromosome abnormalities better. Okay, so consider a case where we had missegregation of the chromosomes in mom, the X chromosome rather in mom, and so that she has produced one gamete which has no X chromosome at all, and another gamete which has 2 X chromosomes. So we have to consider for the individual that's going to be born what the contribution of the father would then be, and there's 2 possibilities for that. The gamete could be fertilized with an X-bearing sperm or for a Y-bearing sperm. So we get different possible outcomes depending on what happens in the dad. Okay, so for example, if you have a female gamete which is missing the X chromosome, and she gets an X-bearing sperm, then that individual has an X chromosome, just one, and that will give rise to Turner syndrome. If she has a gamete with no X chromosome and it's fertilized by a Y-bearing sperm, then that's not viable because you need to have an X chromosome to live. Okay, if you have a case where the gamete from mom has 2 X chromosomes and then gets fertilized by an X-bearing sperm, then you get 3 X's in a child, and that will give you a triple X syndrome. Or if you have 2 X's from mom and you get a Y-bearing sperm, you have XXY, and that's Klinefelter syndrome. Now, that is not the only way in which you can get these chromosome constitutions. You could have missegregation in the dad, for example. This is just one way of illustrating this particular case if this happened. But there are other ways that you could get similar genotypes from other mechanisms having to do with the dad or whatever. So here's a Turner karyotype. She has one X chromosome and no Y. Klinefelter's karyotype. He has two X's and a Y. And in terms of phenotypes, the XO is phenotypically normal. They tend to be very short for reasons that sort of make sense. Puberty fails to occur, but the fact there is ovarian failure early in life, but hormone treatments help, and there's very many treatments that are actually make it possible for XO women to have normal lives. XXY is phenotypically normal, but often infertile. And there's a great range of phenotypes here. Sometimes there's some degree of feminization, maybe slight cognitive in problems, but not much. But there's such a range that goes from completely absolutely normal that you wouldn't if I had a chromosome problem to something where he needs to get some medical attention to, and hormones, of course, help here too. And the triple X females are phenotypically normal. Okay, I think I just wrote this. Okay, but you do have to contrast this into situations where other chromosomes are trisomic or monosomic. Like we don't deal with any monosomies at all. They're lethal except for the X chromosome monosomy, for example. And the XXXXXY trisomies are perfectly fine. So we have to have a mechanism whereby that occurs because it's really abnormal. It doesn't happen for any of the autosomes at all. And that is our dosage compensation system, which we have, which balances the X chromosome transcription between normal males and normal females. Which makes sense. If you think about it, women have two X's and men have just one. And you don't want to have twice as much expression of X-linked genes in females versus males. So in a way to equalize the situation, females inactivate all but one X chromosome in each cell. And that equalizes the expression. It means we have one active X in each cell, which is what men have, which is to say one active X in each cell. So we have to shut off the expression from one of our X's, and this is done early in embryogenesis. Okay, this is an example of epigenetic regulation because the genes that are on that inactive X have been silenced, but they are nonetheless perfectly normal genes. Okay, so as a result of this inactivation process, mammalian females have what's called monoallelic expression of X-linked genes. Okay, even if she's heterozygous, only one allele is active in each cell, but you're going to get some of each kind of cell, as I'll show you in a minute. Okay, so that even though we have two copies of X-linked genes, only one is expressed in any cell, but within the body, both are expressed because of the way the inactivation process works. Okay, so heterochromatin versus euchromatin. Heterochromatin is basically silenced chromatin, which is what the inactive X is going to look like eventually. Okay, it consists of tightly coiled chromatin that doesn't loosen during the cell cycle because transcription is very, very much reduced from this type of chromatin. Okay, so regular chromatin is euchromatin, which changes condensation state during mitosis. The heterochromatin is always darkly staining. It's tightly coiled, although it does, of course, open up for replication and for a certain amount of transcription that occurs from some of the genes. Okay, so the inactive X is heterochromatic and is visible in the nuclei of females. It's called the bar body. If you look at female mammal nuclei, you see this in the nucleus. Okay, so even though we can have an extra X, we can tolerate even more because of this inactivation system. Now, this doesn't work for the autosomes at all. Okay, the bar body is if you're a male, you don't have a bar body because you have only one X and you need to have it active. If you're a female, you've got XX, you have one bar body. Turner females don't have any bar body because they have only one X and it has to be active. XXY males, Klinefelters, they do have a bar body, which inactivates their extra X, leaving them with mostly an XY phenotype, which is pretty close to normal male. Okay, triplo X females have two bar bodies or one large one. The extra one, every extra one is actually inactivated. Okay, there are some genes on the X that escape inactivation, but they're pretty rare, and some are important. Okay, so representing how this kind of works during embryonic development, I'm representing this 6-celled embryo, which is usually is more cells than that, but this is just representational, where I've got a XM, which is, say, maternal, XP paternal, X in each of the cells. So at some point during embryonic development, one of the X's is chosen to be inactivated, and it is inactivated, leaving the other X to be the active cell, active X in that cell. Now this is random as to what X is chosen in each given cell. So you're going to have some cells where the maternal is active and some where you have the paternal one active. Now once inactivation has occurred, all the progeny of that cell have the same one inactivated. So this cell, whenever it divides, it's going to give you an inactive paternal and an active maternal, and vice versa for the ones with the paternal ones inactive. So it remains throughout the life of the female which one is inactive or active in that particular cell lineage. Here's a picture. I don't know where I got this from, but it represents the idea. The paternal X is colored blue, and the maternal one is pink, right? So in the zygote, both are active, and then you have some early divisions, but at some point, it's not really two, it's more cells than that. But at some point, there is random choice of which X is going to become inactivated, and then the process is it becomes inactivated. And then after a while, after that cell proliferates, you're going to have essentially a clone of cells in a patch in the organism that have the same one inactivated. So that's represented by these little blue spots and little pink spots in the final organism. So this is a mosaic. It's considered a mosaic because it's a mixture, a patchwork of different X's active in different cells nearby. Okay, so here's a really good example of a X inactivation in process. This is a female calico cat, and she has some patches of black fur and some patches of orange fur. So the black versus orange is determined by a gene that's located on the X chromosome. So she's heterozygous. She's got one black allele and one orange allele, but they aren't both expressed simultaneously because of X chromosome inactivation. So in some of her cells, the one with the orange allele was inactivated, leaving the one with the black allele active. And so you have this black patch of fur. Then she's got the orange fur, and in those cells, the orange allele is active, and the black one is inactive. So this is what X chromosome inactivation looks at like in terms of the patches that are formed, because once a particular cell inactivates one, all the progeny of that cell keep the same one inactive. Okay, so Barr bodies are darkly stained inactive X chromosomes, visible in the nucleus, and the process of inactivation is called lionization after the woman who discovered it. So you should know about Barr bodies. Okay, so this is a clicker question. Barr bodies are visible evidence of heterochromatin formation. Which of the following is a sex chromosome karyotype of human males with Klinefelter? All right, Klinefelter males are XXY. This is a slightly strange question, but the XYY karyotype can only result from non-disjunction at blank in blank. Well, clearly it can only result from non-disjunction in a male, right? But it turns out it can only occur during meiosis 2. That in meiosis 1, the X and the Y must have separated correctly to give you the two Y chromatids, and that those two failed to segregate at meiosis 2. That's a little hard to think about. So the answer is C. Okay, so I want to talk briefly about the very superficial view of the mechanism for X inactivation. The long non-coding RNAs that are encoded by the X are key here. Okay, so a particular X will either produce what's called the exist RNA, or else it'll produce the antisense RNA, which is T6. So there's a region on the X chromosome called the XIC, X inactivating center, where these and other regulatory RNAs are found. Okay, so this would not occur in real life because you would have either the exist or its antisense expressed, not both. Okay, so what's happening here is the exist RNA is produced from the X inactivation center on the X that's going to be inactivated, or the process of inactivation. So the exist is the thing that actually coats the X chromosome that from which it's made. It doesn't float around in the cytosol, or the nucleoplasm rather. And so it actually physically coats the X chromosome, and that triggers the inactivation process of that X chromosome. So meanwhile, the other X expresses the antisense RNA and remains active, because exist is not expressed from the active X. So you can have a choice which X is active. Here's a sort of model where people think about it. The exist RNA is produced from the X inactivation center, and then it spreads all along that chromosome, the one from which it is made. So as it spreads, it coats the RNA, the chromosome rather, and that recruits things like chromatin remodeling complexes and DNA methylases, etc., that actually cause the chromosome to become inactive, or to actually establish the inactive condition. And that from that point onward, that same X will remain the inactive one. Okay, so we need the regulatory RNAs exist, etc., and the actual mechanism for how the condensation and the recruiting of the remodelers occurs is certainly still being worked out. All right, so it's important that this is a non-coding RNA, doesn't produce a protein at all, and it coats the inactive X, which is the one from which it's made. Okay, so we're mostly going to talk about the X and Y, the X amazing Y chromosome next time, Monday, but I want to point out a couple things right now. Okay, X and Y do not contain the same genes as each other, right? We've got an X and it has its X genes, and the Y is much smaller, has many fewer genes, which are largely not the same as what's on the X. But there is this region where there's par 1 and part 2, the important one being par 1, which actually means pseudo-autosomal region, where the two chromosomes actually are homologous. So within this short region on the end of the Y, there is homology to the same short region on the end of the X. And those two regions are actually able to pair during meiosis, allowing them to correctly recognize who they're supposed to segregate from. So there is a pairing there because of homology. The same genes are located on both. And in fact, there is what's called an obligate crossover. It must occur in that region in order to have the chromosomes stay together correctly and segregate correctly in meiosis. So the two aren't homologous, but they've got enough homology to behave like homologs when they're actually undergoing meiosis. Okay, here's another picture of the pseudo-autosomal region. Okay, so the pseudo-autosomal par 1, they are homologous, side of the obligate crossover, and that enables them to behave as a pair. And in fact, I didn't say this, but it's true that the par region genes generally escape X chromosome inactivation.